Hello, today we're going to be explaining the process of protein synthesis, and this instructional video was created by me, Chase Vuong, and Jacob Yu. Firstly, we have a little helper here, Mr. Piggy, who seems to be very hungry and thirsty. But what is Mr. Piggy craving? Milk, which in this case contains a source of energy that is inside of milk. Carbohydrates, specifically sugars, in this case, lactose. When Mr. Piggy drinks the milk, it travels into his mouth, through the esophagus, into the stomach, and finally reaches the small intestine, where lactose can finally be broken down into enzymes produced by the small intestine called lactase. Lactase, ace being at the end of a word that is usually an enzyme, breaks apart the bonds inside of lactose, creating two new sugars, glucose and galactose. These two new sugars are then absorbed by Mr. Piggy and used for other chemical reactions, such as cellular respiration that turns these sugars into chemical energy. A question still remains: how does Mr. Piggy even get these enzymes in the first place? And is lactase just there in Mr. Piggy's small intestine? Well, this is all due to a process called protein synthesis, which consists of 4 different steps: transcription, RNA processing, translation, and post-translational modification. Firstly, let's discuss the very start of protein synthesis, transcription.
Transcription is a first step in protein synthesis, where a specific segment of DNA is being transcribed into mRNA. DNA in the nucleus is typically stored in a tightly wound form around proteins called histones, forming a structure known as chromatin. The cell must first loosen this tightly packed DNA to expose a specific gene to be transcribed. Multiple factors can initiate epigenetic modifications, but essentially, histone acetyltransferases, HATs, loosen the chromatin. Transcription can now begin, and the DNA is unzipped by an enzyme called RNA polymerase by binding to the promoter region of the DNA. This is done through a process called initiation. Upstream of the transcriptional start point lies a nucleotide sequence containing TATA called the TATA box. Transcription factors, which can recognize the TATA box, bind to the DNA and allow for RNA polymerase to bind in the correct position and orientation. RNA synthesis begins at the starting point and unzips the DNA, creating a transcription bubble. The unzipping and formation of a new mRNA strand is referred to as elongation. During elongation, the enzyme RNA polymerase moves along the template strand of DNA, reading the nucleotide sequence. This is the strand that runs in the 3 to 5' direction. Using this strand as a guide, RNA polymerase builds a complementary strand of RNA to the growing RNA chain in the 5 to 3' direction, while zipping back up the DNA. Transcription ends when RNA polymerase reaches a specific DNA sequence called the terminator. This sequence signals the RNA polymerase to detach from the DNA and release the newly synthesized RNA molecule. This is called pre-mRNA.
Moving on to the second step of protein synthesis, RNA processing takes place inside of the nucleus. RNA processing is a process where the pre-mRNA strand is being modified by numerous different processes. Starting off with the mRNA strand, we can see that there are 2 different ends to this strand, the 5' end and the 3' end. To the 5' end, a process called capping takes place where the guanine nucleotide bases are added to the mRNA strand, creating a cap. Similar to capping, polyadenylation is a process in which adenine bases are added to the 3' end of the mRNA strand, creating a tail. The purpose of both capping and polyadenylation is to protect the RNA from degradation, facilitate its export from the nucleus to the cytoplasm, and ensure proper ribosome binding for efficient translation. Next, a complex of RNA and proteins, spliceosomes, come to the mRNA and begin to cut segments of the mRNA that don't code for lactase or the protein that is going to be synthesized. The part of the mRNA that just got cut out are called introns. The parts of the RNA that do code for lactase in this case, or for the protein being synthesized, are called exons, and they are joined together with the help of the spliceosome. This part of the process is called RNA splicing. But what if I wanted a different protein other than lactase, or wanted to change the structure of lactase? This is why there's alternative splicing. Alternative splicing is a combination of different introns and extrons that each impact the protein being synthesized by changing the domain or structure or function of the protein being synthesized. Let's say I wanted to change the binding sites of this enzyme. With unique combinations of RNA splicing that can make different proteins or change the structure of the enzyme being synthesized that result in the function of this enzyme being changed. It's important to remember that the structure of a protein impacts its function. similar to the binding sites being changed here, being able to bind to different substrates from the original enzyme and change the domain of this enzyme. Now that the mRNA has been fully modified, it can move on to the next step of protein synthesis by escaping through the nucleus's nuclear pores and into the cytoplasm. Moving on to the third step of protein synthesis, translation. Translation occurs in the cytoplasm where the mRNA strand reaches a ribosome. Ribosomes have their own ribosomal RNA, or rRNA, that allow it to move across the mRNA strands. Additionally, ribosomes consist of two subunits that attach to the top and bottom of the mRNA strand. But before we can explain any further, let's discuss nucleotide pairing briefly. In RNA, the nucleotides U, uracil, and A, adenine, are both complementary to each other, while in DNA, the nucleotides T, thymine, and A, adenine, are both complementary to each other. However, G, guanine, and C, cytosine, are both the same in DNA and RNA. Moving on, the ribosome consists of three different segments that each contain different functions. E, the exit area, where tRNA will leave the ribosome after it finishes adding its amino acid to the polypeptide chain. P, the part where tRNA and its amino acid are bonded to each other in the polypeptide chain. And A, the tRNA being added on top of the mRNA with its amino acid. The way that tRNA functions is that it reads 3 nucleotides at a time from the mRNA strand. These 3 nucleotides are called a codon. In this case, A, U, G are all part of one codon. Once tRNA reads the codon, it creates complementary bases to the codon, creating an anticodon. In this situation, U is paired with A is paired with U, and C is paired with G. The amino acid based on the codon chart would be MET. MET is an amino acid that marks the start of translation, making it the only start codon. That explains how tRNA is able to be paired with the mRNA, but how does the tRNA get its amino acids? Well, that's the purpose of amino-acyl tRNA synthesase. It is a protein, specifically an enzyme, that has 2 separate binding sites, one for the tRNA and one for the amino acid specific to the codon that it will read. With the help of this enzyme and powered by ATP, the amino acid and tRNA are both bonded together and can now move on to the A section of the ribosome. So since the ribosome already read the first codon, it moves on to the next, shifting the tRNA with the MET amino acid into the P section of the ribosome, and a new tRNA with the new amino acid is set in the A section of the ribosome. Now the ribosome moves on to the next codon, placing the tRNA that doesn't have an amino acid anymore in the E section of the ribosome, and shifting the tRNA with the PHE amino acid into the P section. That binds the 2 amino acids to form a bond. This bond is called a peptide bond, and a new tRNA with new amino acids moves to the A section of the ribosome. Then the tRNA currently in the E section of the ribosome is ejected back into the cytoplasm. This process repeats continuously until it reaches one of the 3 stop codons that signal the end of translation and forces the polypeptide chain to be released from the ribosome and is left in the cytoplasm.
After the lactase polypeptide is synthesized by ribosomes, it begins post-translational modification. It enters the endoplasmic reticulum where sugar groups are added to specific asparagine residues of lactase. This helps the protein fold correctly and become more stable. Chaperone proteins like calnexin and calreticulin assist in proper folding of the enzyme's 3D structure. Misfolded lactase is targeted for degradation in ER-associated degradation or ERAD. Meanwhile, properly folded lactase is transported from the ER to the Golgi apparatus. In the Golgi or intestinal lumen, prolactase undergoes proteolytic cleavages. Enzymes like furin or related proteases cleave the prolactase to produce the mature active form. The active lactase is packaged into vesicles and transported to the apical membrane of intestinal cells. Lactase is finally anchored to the brush border via its transmembrane domain. The enzymatic portion is exposed to intestinal lumen for lactose hydrolysis. Lactase can now do its job.
Now that Mr. Piggy has a better understanding of protein synthesis, Mr. Piggy knows that humans with lactose intolerance don't have much or any lactase inside of their bodies. However, Mr. Piggy and everyone that has a high concentration of lactase inside of their bodies is lactose tolerant. Sadly, Mr. Piggy has to go, and so do we. We thank you for watching this video and hope you learned something new.